concern associated with cannabis and other substance use is the effect it has on the developing brain, particularly in young children and adolescents. Unfortunately, the vast majority of research in this region and area has really been done cross-sectionally. The Adolescent Brain Cognitive Development Project presents a massive investment by NIH to really look at this question longitudinally. And so today we're really pleased to have Dr. Alex Miller here to present some of the early findings from this program. I'll let him describe ABCD in detail. But Alex Miller is an assistant professor of psychiatry at Indiana University School of Medicine. He completed his Ph.D. at the University of Missouri and a postdoctoral fellowship in substance use disorder genetics at Washington University School of Medicine. His research, which was supported by a K01 by NIAAA, focuses on applying statistical genetics and neuroimaging data analysis to better understand the developmental neurogenetic etiology of substance use disorders and impulsive personality traits as risk factors for substance use disorder onset and progression. We're very pleased to have Dr. Miller with us, and Alex, it is all yours.
Okay, thank you guys for having me. Pull up my slides here.
Oh, I will just note too that this meeting is being recorded, so just keep that in mind when you make your comments.
Everybody hear me okay and see the slides?
Yes.
Okay.
Good to go.
So yeah, today I'm going to, my presentation is on some of these neuroanatomical differences that are, associated with substance involvement, and we used kind of, the ABCD study to ask whether, some of these differences we've seen across the years in brains associated with substance involvement, whether they are to some extent predispositional, so whether they're evident prior to any substance use initiation in kids in this study. So my effort on this project was originally funded with a training grant from NIDA, and then more recently a K01 from NIAAA, and I have no other conflicts to disclose. So I wanted to start first by talking a little bit about the theoretical models of brain substance use relations, and kind of the predominant model, I think, for the last couple decades at least, focuses more on the process of the development of addiction itself, and really emphasizes the brain's role in this as consequential to the development of addiction, such that prolonged exposure to substances and developing an addiction really causes brain changes in connectivity or structure that sort of begets this cycle and kind of perpetuates addiction. But more recently, contemporary models have kind of also focused on how genetic risk, in particular, but also environmental factors, may influence changes in the brain that are predisposing to substance use. So over here on the side, I kind of have a model where, let me make my laser if I can. Okay. So this model sort of follows, in general, the addiction model, the kind of predominant model that potentially genetic variations or environmental factors influence one to start using substances, and then heavy prolonged exposure results in neurotoxic effects in the brain. And here I just mean changes in the brain, essentially, that kind of beget potentially more substance use. But there's also the possibility that environment and genetic influences sort of directly influence brain development to some degree, which may predispose individuals to have a higher likelihood for initiating substance use, which then also kind of on the consequential end leads to these neurotoxic effects with more changes on brain development and more likely to use substances. And so as Tom mentioned earlier, typically, the interpretation of brain substance use relations that we see in research are interpreted in terms of consequences that are consistent with the neurodevelopmental model of addiction. And substance involvement here kind of broadly defined, which can mean a lot of things, obviously, as early as just experimentation or early initiation to substance use disorders, are generally associated with lower gray matter volume across the cortex, thinner cortical regions, and less white matter integrity. But historically, at least, a lot of these studies have been conducted using smaller cross-sectional samples, though there are certainly some exceptions. And as I stated, that these are largely interpreted in the context of this predominant theory, that prolonged substance use alters brain structure and function. And over on the right here, I have an example. This is not a small study by any means, but this was a meta-analysis of different substance use disorders. Here, I'm just showing alcohol use disorder. But I think, following the publication of this paper, a lot of the interpretations, not necessarily by the authors, but were that these are these differences are resulting from substance use disorder. And so another example of this kind of brain changes resulting from substance use that is particularly a little more applicable to initiation, which I will be talking about mostly today. So this also not a particularly small study, but around 800 adolescents, where they looked at participants who are cannabis naive at baseline, which is around the age of 14, and then they looked at follow-up scans. So I had a scan at baseline and a scan at this follow-up at age 19, and looked to see how brain changes might be related to cannabis use in the intervening period. And what they found was that the prefrontal cortex was thinner in those who had initiated cannabis in between the baseline scan and the follow-up scan, and that there was a thinning effect, so that the cortexes were thinner in the prefrontal regions at follow-up after initiating cannabis use, and that there was a dose-response relationship in this thinning. So the more these adolescents had used cannabis lifetime at this follow-up, the thinner their cortex was relative to their baseline. And this thinning was also associated with greater attentional impulsivity at this follow-up. And then they conducted a follow-up analysis of these results, essentially looking at this through more of a causal network. And their results suggested that indeed that this was potentially causal in nature, that this onset of cannabis use may have resulted in thinner cortexes at this follow-up. So then on the other, and these are just examples, there's a lot of papers that look at these. This is another example where potentially the opposite is true, where we have alcohol naive participants at baseline, and we look at scans in terms of their baseline scans and their probability of initiating alcohol at a follow-up. And so this is essentially the same region of the brain where we see lower prefrontal cortex volume is associated with an earlier onset of alcohol use relative to kind of a higher or even a potentially more normative prefrontal cortical volume. And so then, at least, for this paper, I'm kind of focusing on early initiation as a risk factor. One, because this is typically one's first experience with substances occurs in adolescence or early adulthood. So this is a time when the brain is still developing and still maturing. And early substance use, early initiation of substance use has been shown to be really associated with escalating use of that substance, use of multiple substances, higher risk of developing substance use disorders in subsequent years. So at the top here, this is alcohol. We can see that the earlier somebody initiates alcohol use, the more likely they are to have alcohol use disorder faster, essentially. And same is true with cannabis. Starting cannabis at 15 or younger kind of confers an increased risk in the likelihood of lifetime onset of cannabis use disorder. And aside from substance use disorder development, early onset of substance use is also associated with other adverse life outcomes, potentially like reduced educational attainment, so potentially school dropout, and just general elevated rates of psychopathology. So there's kind of converging evidence, I think, that suggests that there are both potentially causal effects, so that the substance used or may cause some of these changes in the brain, but there's also evidence of predisposition or differences in these brain structures prior to initiation makes initiation more likely. And there's kind of a mix. This is a review for alcohol involvement specifically, but you can see that relevant to the previous two studies that I talked about, that reduced volume or thickness of this prefrontal region both exhibits potentially causal associations or predispositional. And so this is in line with kind of the model that I was talking about earlier, where predispositional risk for substance use may be related to different trajectories of brain development. So lower volumes, potentially, like in this case, the prefrontal cortex is associated with more predispositional risk. And then at the same time, once substance use onsets, particularly chronic substance use, this may lead to further brain reductions that are associated with heavier use. And at the same time, as I mentioned earlier, we know that some of these earlier, smaller cross-sectional studies may have overestimated the effect sizes that we should really be looking for. And as sample sizes have grown, we've come to realize that brain behavior associations are really probably characterized by fairly small effect sizes. Somewhere in the neighborhood of a correlation of an absolute correlation of 0.1 or smaller. And then as you increase sample sizes, the correlation kind of converges to a more precise estimate around this small effect size, whereas in these earlier, smaller cross-sectional samples, we see really large, potentially effect sizes that maybe have been overestimated to some degree. So that brings me to the study that I want to talk about today that was just published in JAMA Network Open, where we used the ABCD study to very basically ask, what are the neuroanatomical features that are associated with early substance use initiation? And to what degree do some of these associations or these brain differences seem to precede initiation? And so I'm at IU School of Medicine now, but most of this research was conducted while I was a postdoc working with Ryan Bogdan and Arpana Agrawal at WashU School of Medicine. So what we did, we used data from the ABCD study, which is a study of almost 12,000 children across 21 sites in the US that are being followed continuously from their baseline at ages 9 to 10 to ultimately 19, 20, or potentially beyond. They go through a series of assessments every 6 months to a year to 2 years, which includes psychological assessments and substance use assessments and environmental assessments of their family and neighborhoods, but also they undergo brain scans every 2 years, and there's also genetic data for these individuals. So it's a really rich database in terms of its size and number of variables and representation of kind of a developing cohort of children in the United States. And so for the current study, we used just under 10,000 of these participants. And basically, we defined some of them as substance use initiators, which turned out to be about 35% of this sample, so a little under 3,500 participants. And the way we defined substance use initiation was any endorsement of really any kind of substance use from baseline through their 3-year follow-up. And that included yearly assessments, but also mid-year assessments of their substance use. And at least in terms of the paper and the available data, this really came out to be any substance use initiation prior to the age 15. And again, when I say any substance use, for a lot of these participants, actually, we're talking about the very earliest stages of experimentation with substances. So just sips of alcohol or puffs of nicotine or cannabis. And then in the contrast, we had a little over 6,000 participants that were substance naive. So these participants at no point endorsed any kind of substance use. With a couple caveats, we reduced this sample based on missingness from follow-up 3. So there were some individuals who endorsed no substance use, but didn't have follow-up 3 data. And so those individuals were not included in this analysis, essentially to protect against possible misclassification, since we don't know whether they had initiated substance use at follow-up 3. And then we removed individuals who only endorsed substance use, particularly for alcohol or nicotine, solely in the context of religious ceremonies. So the major comparisons of the paper are essentially just comparing substance use initiation versus substance use naive children in the study. And so to do this, we looked at baseline brain scans, which, based on some of the literature earlier, there are some regions that are kind of, I think, coming together with converging evidence that these might be regions of interest. But based on the sparse data, we had sort of pretty low priors on what regions might be involved. And so we took more of a brain-wide association approach. And so we looked at global metrics, we looked at subcortical structures, particularly volumes, and then we looked at the cortex across 4 different modalities. We looked at volume of the cortex regionally, we looked at thickness, surface area, and sulcal depth of these regions. So in all, we looked at 297 different brain phenotypes. And our primary contrast was between substance use and no substance use, but we also then conducted some follow-up tests looking at alcohol, nicotine, and cannabis separately. And so our analytic models involved mixed effects regressions, where we included some fixed effects covariates, particularly age squared, participant sex, their pubertal status at baseline. Then we accounted for family relationships, both as a random effect where we nested family within sight, but also family relationships that were not captured by overall family ID, like sibling, twin, or triplet status. And then because the data collection takes place over many sites with different MRI scanner models, we included MRI scanner model to account for any variability there in the imaging acquisition. And then, as I mentioned, we looked at global metrics, but then we looked at regional metrics as well. And when we did that, we included the global kind of comparative as a covariate. So when we looked at regional cortical thickness, we included the global metric of cortical thickness. So these are measures of the regional cortical thickness after accounting for differences in entire thickness of the cortex. And then because we were looking across so many different regions, we wanted to kind of balance out this exploratory approach in terms of protecting against both false negatives and false positives. So we corrected our tests both using a study-wide Bonferroni correction, which was basically a Bonferroni correction for almost 1,200 tests that we conducted. and then we did FDR correction within substances. And then the aspect of the study that really gets at whether or not some of these differences precede initiation included follow-up analyses that controlled for prenatal exposure, so adding that as an additional covariate, and then excluding participants with initiation prior to or at baseline. So we so here's sort of a schematic that hopefully is helpful in kind of understanding what we did. But here we have all of the substance use assessments from baseline to the 3-year follow-up. And we have our brain scans at baseline. And so our overarching question, kind of our correlational question, was how do those who endorse substance use at this point differ from those who don't based on their baseline scans? And then to get at the potentially predispositional aspect of this, we reduced the sample to those who had just initiated after baseline. So we have baseline scans associated with whether or not after that scan somebody endorses substance use initiation. So this is just a breakdown of kind of what the substance use initiation looked like in this study at this point. And something that you'll notice is that most of this substance use was alcohol, and most of that was sips. So we have about, I think it's like 90%, it's on my next slide, how much of this substance use was alcohol. And we have smaller samples for nicotine, which was 431, and cannabis, which was 212. But also the proportion of these individuals who had already endorsed this substance use at baseline was much different in terms of alcohol to the other substances. And again, this is potentially related to SIPs being part of our phenotype here, but you can see, at least for cannabis, only 11 participants had endorsed using cannabis at baseline, and that climbed pretty dramatically by follow-up 3 to 212, whereas we have about 2,000 endorsing use of alcohol at baseline, which climbed to 3,000. Also noting that for cannabis and nicotine, there's considerable overlap with alcohol in terms of using both, or in this case, using all 3. Just because this is interesting to me, mostly, sort of buried in the supplement, but we also sort of a little bit characterized some of the methods or some of the ways kids were using substance use initiation at this substances at this stage. So like I said, alcohol was most of our substance using participants, and most of those were SIPs, with kids endorsing full drinks only being about 192. Cannabis, the predominant route of administration, was smoking flower or bud, but almost half also endorsed consuming edibles. And then interesting for nicotine, the majority of participants in this category endorsed using e-cigarettes or vaping, with 60% of them endorsing that exclusively, and less than 1 in 5 endorsing using traditional tobacco cigarettes. And so in addition to these 3 main substances, there was also 213 participants who endorsed other substances as well, and about half of these only endorsed other substances. So those include things like inhalants or stimulants, or using over-the-counter medications recreationally, things like that. So moving on to some of the results of the study, and I'm focusing here first on the global brain differences, and then I'll kind of break down regional brain differences by substances in the next few slides. And these mostly are reflective of any substance use or alcohol use, and we didn't see any significant differences in nicotine or cannabis, and part of that may be the smaller sample sizes that were involved in that contrast. But here we see really larger global metrics. So greater whole brain volume, total cortical volume, intracranial volume, greater total surface area of the cortex, and then greater total subcortical gray matter volume. And we can see that these effect sizes, again, sort of line up below this 0.1. So these effect sizes are really kind of what recent research has led us to conclude or what's to be expected from these brain behavior associations in larger sample sizes like this. So then regionally, these were all Bonferroni significant. So these were very significant findings in terms of their p-values. Moving regionally, we looked at any substance use. In addition to the global findings, there were 3 regions that exhibited this Bonferroni significance threshold. And that was thinner rostral middle frontal cortex, a thicker lingual cortical region, and then a larger lateral occipital volume. And so all of these slides, I'm essentially just showing the brains here. The bold outlines are regions that were FDR significant, while these gold ones are the ones that were Bonferroni significant. So a little bit stronger evidence in terms of their association with substance use initiation. And then above is just a plot broken down by regions of the cortex and the log p-value. So the higher number here is a lower p-value, and just highlighting these 3 that survived this Bonferroni correction. And then we had many more that survived this FDR correction in terms of regions in the cortex and subcortical structures. And we noticed a pattern really of terms of cortical thickness. So most of these associations were with cortical thickness. And then the pattern was that we're seeing less thickness in frontal regions. So we can see that here in blue. These are negative associations across these brains. But where we see significant associations with cortical thickness in other regions of the cortex, it's actually thicker. So there's kind of this balance or sort of imbalance pattern where we have thinner prefrontal cortexes and thicker cortexes elsewhere. And then in terms of subcortical structures, we see a larger bilateral globus pallidus and left hippocampus with any substance use initiation. And then for alcohol use initiation, which, based on previous slides, there's a large overlap between these 2 contrasts. So the results really largely mirror the any, but we're actually seeing more regions kind of come out at a stronger level of evidence, and even more of this pattern in terms of thinner cortex of the frontal regions and thicker cortex elsewhere where things are significant. And again, we see a larger global pallidus in these analyses. So then results are a little less exciting, I guess, for nicotine and cannabis use initiation. And again, with the caveat that these are much smaller samples and potentially smaller samples than we need to make these contrasts in terms of power and expected effect sizes. But for nicotine initiation, there were 2 regions that exhibited FDR significant associations. One was smaller volume of the superior right frontal gyrus, so here, and then deeper left lateral sulci of the orbital frontal region, so down here. And then with cannabis use initiation, we see a similar number of regions, really 3. One being smaller right inferior parietal volume, so that's over here. And then a smaller right caudate volume, which is kind of in contrast to the results we see for any substance use and alcohol substance use, and also the global tests of subcortical structural volume, where we see larger volumes. here we're seeing a smaller volume of the right caudate for cannabis use initiation. And then we're also seeing a thinner left precentral gyrus. So then moving to these additional analyses, where we kind of tried to tease apart some of these correlational aspects to say a little more about the degree to which some of these findings may reflect predispositional associations. So first, we included prenatal exposure as a covariate in these models, and that was both substance specific. So for alcohol initiation, we included potential prenatal alcohol exposure as a covariate, but then we looked at also any prenatal exposure across all of the substances. And essentially all of our findings were robust to inclusion of this covariate, suggesting that these associations are independent of whether or not these kids have been exposed prenatally to any of these substances. And then in the models where we restricted to just post-baseline initiation, we had a pretty sizable drop in power, at least for any substance use, and for alcohol, less so for cannabis and nicotine, just based on the rates of baseline initiation. And what we saw was that the majority of these bonferroni, so kind of our strongest associations, remain significant after, moving to this post-baseline initiation model. And largely this reflected the global findings, which were still significant after post-baseline sample restriction. And then for alcohol, we see even a greater number where 76% of these associations were still significant, which included this thinner rostral middle frontal gyrus. And then for nicotine and cannabis, all of the results that I presented on were significant after reducing to this post-baseline initiation model. And then we ran some tests essentially between the effect sizes that we're seeing for the whole sample versus this reduced sample. And in general, that's over here, broken down by regions and metrics and substances. We see a pretty strong concordance between the effect sizes that we're observing in the full sample and those that we're observing after reducing to post-baseline initiation. So the main takeaways, I think, of the study are the findings of larger global metrics and subcortical volumes, which is somewhat inconsistent, at least when we look at the literature in terms of substance use disorders and these types of metrics, where we really generally see lower, lower whole brain volume, lower subcortical volumes, with the exception here for this trend in our results being the smaller caudate for cannabis. And then this interesting thinner prefrontal cortex, but thicker cortex elsewhere. At least the thinner prefrontal cortex is pretty consistent with other studies looking both at initiation, looking at it predispositionally and causally, and looking at just initiation, but also substance use disorders. So thinner prefrontal cortex is pretty consistent with those findings and may kind of represent a brain phenotype that kind of is consistent across development with initiation of use and developing substance use disorders. So it may be, again, one of those regions that is both potentially predispositional and consequential to using substances. All of these differences, like I said, were evident after accounting for prenatal exposure. So really getting at truly substance naive participants here, and that most or many of these were evident prior to initiation. And then again, the small effects which were expected are what we see really. And so linking some of these findings a little bit to some of the research I'm interested in doing and some of the research that others have done, and sort of connecting these pathways between our genetics and our brain structures and potentially impulsivity as a developmentally relevant phenotype for the onset of substance use. So we've seen shared genetic influences between alcohol consumption and sensation seeking with greater surface area of the cortex, which is somewhat consistent with our finding here of initiation being related to greater surface area. But when we also look at alcohol consumption and AUD in terms of their overlap in genetic correlations with cortical thickness, we also see this decreased thickness of the PFC, suggesting that there may be some genetic kind of pathway or link between alcohol consumption, alcohol use disorder, so substance use and substance use disorder development with this thickness of the PFC. And then other studies, I mentioned the globus pallidus earlier as being one that kind of came through as larger, as a specific subcortical structure that was larger in those initiating. And this is consistent with some previous ABCD studies where larger global pallidus has been linked to sensation seeking both at baseline and at follow-up too. And then a recent study looking at cannabis use disorder genetic risk and its effect on adolescent cannabis use, and finding that there's some level of mediation of that genetic effect on adolescent cannabis use by both novelty seeking and lower white matter integrity in the brain. So in my mind, some of these things are really, I think, starting to come together and getting kind of a broader picture of the relationship between our genetics, our brain, some of these impulsive personality traits that predispose to substance use involvement, and then progression of that substance use. Some limitations. I've listed a lot here to be comprehensive, but this is just initiation, and in this case, it was fairly broadly defined. So comparisons with other forms or other phenotypes of substance involvement at this stage, I think, are a little tricky. And as these kids age, we'll have better, more kind of focused phenotypes of the type of substance use they're engaging in. As I mentioned, because we had sort of poor priors about what regions might be involved in this initiation, and we wanted to be overly inclusive, we included many brain features, but that kind of results in us using multiple test corrections that may have resulted in false negatives potentially. I've already mentioned a few times the small effect sizes, but I think it's important at this stage, given that, with these types of effect sizes, these findings are not directly clinically relevant at this stage. But I think they're very valuable for understanding the etiology as we kind of try and move forward and tease this apart a little bit more. But potentially there's the possibility that using multivariate approaches, so not just focusing on one region versus another region, but maybe a collection of regions or a network of regions may result in larger effect sizes in these types of comparisons. We only focused on the baseline, and so we didn't really evaluate change in terms of baseline to follow up to in brains, or differences from baseline to this year 2 scan in terms of post-exposure. We considered looking at that, but the rates of endorsement between those two time periods, between baseline and 2 years in, results in smaller sample sizes. And so it's not as strong a test, I think, at this stage of post-exposure. And with the caveat that this could also be predispositional to some degree. Your change from baseline to follow-up to may be influenced by environment or genetics. And I mentioned, cannabis and nicotine being smaller, maybe on the lower end of being sufficiently powered for the effect sizes that we're seeing. And then really, there's a lot of other factors that may be at play that we either didn't measure or can't measure at this stage, but definitely want to follow up on some of those things. So I really see these limitations as opportunities for follow-up. in our kind of upcoming research. And so what we want to do really is now look at some of the sources of variation in these brain structure differences. So maybe, these brain differences are related to or are predispositional for substance use initiation, but what sorts of factors cause these brain differences in the first place, and at what point do these brain differences start to emerge? And so that might involve looking at mediation models that try to tease apart the degree to which environment, including, socioeconomic status or social determinants of health, neighborhood variables, things like that. To what degree are their influences on substance use initiation mediated by some of these brain differences? And researchers have already, with this data set, even started to look at questions like this. So a recent publication looked at brain connectivity, and they found that resting state connectivity that's related to accelerated maturation predicted substance use initiation in this sample and is associated with pollution. So sort of getting at what are some of the environmental influences on these brain differences. And then we also want to look at what are some of the genetic influences on these brain differences. So looking at polygenic scores and conducting, ABCD has a sizable proportion of the sample that are twins and or siblings. And so looking at potential genetic influences through these models as well. And then, again, as these kids start to age and start to use more substances, inevitably, longitudinal analyses will be important to sort of evaluate the degree to which these differences we're seeing now, whether those are the same differences that we might see with later stages of substance use in the sample. With, again, the caveat that without sort of controlling for genetic influences, to some degree, these could also reflect predisposition. So I want to thank my co-authors and funders for the research, and the ABCD study for letting us take a look at this data. And then I'll kind of open it up to questions if anybody has any, or discussion.
Thank you, Alex. That was fantastic. Very careful work, and you did a terrific job of explaining it all to us. I open it up to questions. You want to just raise your hand? We have about 40 people attending. So yes, Edith.
Um, I was wondering, does the ABCD database have really good phenotypes that are related to impulsivity so that you could evaluate the